Thank you very much for the nice words, and I also would like to forward my thanks to the audience who was waiting and who is going to join me for the reflections of my previous work and also on my current work. By preparing this talk, I was really overwhelmed of the work which has been done in more than 4 decades now, and you may understand it's basically impossible to talk about everything that has been done by me and my colleagues. And therefore, I would just like to focus on some very interesting aspects of the beginning of my work and then go on to the present problems and questions we have. And so the first ones are here marked in white, and the later, the current activities are in yellow, and there's lots of work done in between, which I'm unable to comment on. I probably have to write a book, or maybe I should take this decision once I'm in Kiel. So the early times, they started with this picture, which was given to me by my supervisor. It's on the replication of bacteriophage FD, infecting E. coli. So already dealing with infectious disease. So it comes into cells as a single-stranded circular DNA. It's made double-stranded, and then it's progressing and going into rolling circle mode to produce a lot of circular progenies. And my task was to do this in vitro and to really find out what actually is the trigger of the replication of this double-stranded DNA that was unknown. And it was presumed it was the so-called gene 2 protein. There are about 10 genes, and this is a gene 2 encoded protein. So I succeeded to purify this, partial purification, full purification. I also made an overproducer and was able in a big fermenter, 100 liters, to purify this enzyme. And together with Heinz Schaller of the university and Klaus Geider, my supervisor, we were able to demonstrate that this protein cleaves the double-stranded DNA at a very specific site and thereby initiates replication. And by doing this and putting some other enzymes into the in vitro tube, we were able to replicate this double-stranded DNA and produce lots of progeny circles. And that's how it ended up. So it was actually quite an interesting time. It was not possible to do PCR. This was discovered in 1983. However, we had at this time 4 enzymes. So the gene 2 protein, which initiated the replication. We also required a helicase to separate the 2 strands, DNA binding protein, and a potent polymerase, polymerase 3. So this took a little bit longer than my thesis, which was very short actually, and was concluded also as a postdoc, and as an early postdoc. Then I simultaneously went to United States to start with something which was, to my understanding, much closer to the clinical problems at this stage already. So working with Neisseria, which is a really infectious agent infecting humans. And here we started to characterize a very interesting phenomenon, namely anagenic variation. This was done together with Magdalena So. So just to introduce you into the infection process of these bacteria, the gonococci and the meningococci, they're quite related. They produce pili, and these pili are adherence factors which recognize a specific receptor on human cells. It's very specific for humans. Unfortunately, despite many efforts also of other groups, the receptor is not really well known, but it is absolutely human specific. So after this attachment, the pili retract and then the bacteria get into close contact, and eventually, if they like, depending on some features, they can invade and transcytose these cells to interact also with phagocytic cells and also with endothelial cells if they, once these meningococci in particular, get into the bloodstream. So it's an interesting model which we try to understand, particularly with a phenomenon which was called anagenic variation. And that affected the pili, which are very exposed structures and therefore prone to be a target by the immune system. So I tried, and this was the first gene actually ever cloned from these bacteria at those times. Fantastic progress already. And once you took this gene for the pili and hybridized it to the genome, we found a strong surprise. Lots of genes related to this pilus gene, and we didn't know what actually is going on. However, when we cultured the bacteria in vitro on plates, some of these loci changed their sizes, and there was a lot of movement and changes going on in the genome of these bacteria. So it took us a couple of years, then also back in Heidelberg and also then in Tübingen, to understand the genomic structure. So there are multiple loci in the genome, some of which turned out to be actually one or two to be expressed, and many to carry silent encoding information, coding for variant parts of the pilin gene. And we also came up with more clear hybridization patterns. And so showing here the same strain, but different variants after cultivation in vitro. And using a specific probe of this particular locus, we found that suddenly sequence information from this locus was transported into this expressed site here. So this was actually the first indication of gene conversion in a bacterium, and one of the first actually in microbes, and also at the same time when gene conversion was discovered in a human genome. So this is what happened. We divided actually the pilin gene in so-called mini cassettes, and it's only fractions from silent loci were copied and used to convert the expressed gene copy. And that ended up in a variant gene copy. So this was the principle. This is an old slide which I just copied while closing the laboratory in Berlin. So indicating once more that these many silent sequences, which contain variant sequence information, which are recombined by the expressed with the expressed locus in a reggae-dependent fashion, leading to altered genes. And this can happen by various pathways. It can be reciprocals or back and forth. It can be non-reciprocals or in one direction. And it can also be transformation mediated. That's a very interesting phenomenon, which we also went into. However, it doesn't matter at this point. The interesting is that by calculating the number of variants that could be potentially produced is about 10 million variant pilins from a single bacterium. And that explains why pilins cannot really be used as a vaccine. It was tried for several decades to use this as a vaccine target, but so far we still don't have a vaccine against gonorrhea. So this is the pili, what they do. They form these microcolonies on epithelial cells, and actually there is a protein in the host cell which prevents that these bacteria penetrate. This is actually caveolin. So when this is expressed, the bacteria stay outside and form these nice microcolonies. However, then depending on whether the so-called opacity proteins from the outer membrane are produced, the bacteria can penetrate into the cells. And this is actually shown here, the expression of these proteins is also variable. So this is a single isolate. After passaging in vitro, you can produce a lot of different of these variants. Again, we have a variable surface of these proteins. Now, this is a different mechanism. In these cases, we have multiple intact copies in the genome, and they can be turned on and off independently. And we wondered how this might work. And by this way, we discovered a highly repetitive sequence, which is actually coding for the protein, and it's a pentameric repeat. And this, with the number of repeat units, turned out to be actually variable, and by this way, shift the gene out of frame, in frame, out of frame, depending on the number of repeat units. So this was actually the first evidence of slip strand mispairing in bacteria, and amongst the first, so at the similar time, slip strand mispairing was discovered also for Huntington disease. In this case, we have triplet codons, which are variable, so not shifting the reading frame, but shifting only the length of the protein. Here we have an on-off switch. Now, in this case, we found that the heterogeneity produced is not in order to serve as an immune escape mechanism, but rather to help these bacteria to interact with different host cells. And we also identified the receptors, so some of these proteins could interact with syndicams, others with CCAM molecules. They are called also previously CD60A and so on. And depending on what protein is produced, the bacteria took different approaches to infect host cells. We also looked at secreted factors. One interesting observation was the identification of the IgA protease, which is a secreted factor cleaving IgA. And we were interested in how this protein could be secreted. It's shown here. It's an autotransporter mechanism. And subsequently, people used this also to present determinants here, immunoglobulin change on the surface of bacteria, which could then recognize other antigens. So this is actually quite nice. And people use this also commercially right now. And we are actually putting here on the surface of bacteria also epitopes of the current coronavirus. So we are a little bit engaged in coronavirus vaccine research, but this is only a side aspect of the current work. We also found, for example, that this IgA protease gene is subject to horizontal exchange. So information coming from outside changes this gene all the time, providing a diversity of these bacteria in the population of these strains. Okay, so let's stop with the former past, and let's jump over of much work with many pathogens, chlamydia, influenza, other viruses, with vaccines, drugs, and host receptors, and numerous other aspects of infection. And let's go into the more recent aspects of our work, namely the question, what is the fate of host cells after infection? And there it appeared that Helicobacter pylori was a very good model. It was discovered only in 1982 by Warren and Marshall. And we started about 8 to 10 years later to work on this bacterium. And we were excited in particular that it was reported to be connected with gastric cancer. Now, this bacterium is associated with about 50% of the world's population. And only 1 to 2% of the infected people can develop cancer, gastric cancer. So this leads still to the fact that Helicobacter pylori is responsible for probably 80% or so There are different calculations reaching from 70 to 100% of gastric cancers, with many deaths worldwide per year. 5 years survival prognosis is really poor, and so it's worth to at least understand the problem, this association of the bacterium with the host and the stomach, and perhaps also take measures against it. So far, we have to say, unfortunately, vaccines do not work. One can cure the infection nicely with antibiotics, but otherwise, the infection stays lifelong. So the target of this bacterium is the human epithelium of the stomach. And this is shown here. This is actually the epithelium in vitro. And we have to accept that the epithelium is not only for Helicobacter pylori, but for many pathogens, the center cord of the contact of the bacterium with the host. It's the center cord of infection, and it's also a center cord of defense. And it is also a target of damage. This is where damage can play a role. Mutations can occur, and these mutations somehow may eventually lead to cancer. So this is the system, and we thought in order to study this, somehow we have to do this with primary human material, because it's a human pathogen, and we should not use transformed cells, which are already transformed, if we want to study cancerogenesis. So we set up to establish the organoid model of the stomach. This was mainly guided by Felix Lehrmann and also Sina Bartfeld. When we took tissue from the human stomach, which we got from surgeries, and generated organoids. They're nicely growing under certain conditions. So basically, these are conditions supporting the survival and the growth of stem cells, adult stem cells in these tissues, as have previously been defined by the group of Hans Clevers and other developmental biologists. So we obtained at a relative early time point in 2014, we obtained these organoids. However, these are, you have to understand, they're like balls. And it's the apical side, so the surface of these cells is pointing to the inside. And there's a lot of mucus secreted, and it's actually quite hard to infect these organoids with bacteria. So we thought maybe we should we should somehow bring this into a linear fashion. And this is what Francesco Bortolato succeeded with. We put this onto an air liquid phase model, which provided highly polarized epithelial cells. So this is actually a natural epithelium from the in vivo, from the gland, from humans. And when you compare this with the mucosoids, which we produce in vitro, so it's basically identical and a very good model to study. This graph is just showing the very high, nice polarization with tight junctions and so on, and basically all the features we can expect from the epithelium of the stomach. It can also be differentiated in vitro by the addition of Wnt and R-spondin, which are important for the growth, for the indefinite growth of these organoids. And if this is removed, the organoids start to differentiate and they start to produce another mucin 5AC instead of mucin 6, and some other markers that are differentiating and actually at some stage also replicating quite fast. This is the rapid proliferating compartment, as you can see here. So this is the gastric glands, and so this tissue actually comprises the more tip-located gland part, and this is the bottom part of the gland, which is reproduced in the presence of wind and are sponding. So having this model at hand, we now investigated a lot of features of this bacterium, and in particular the aspects of inflammation, which was a hallmark of this bacterial infection in the stomach. And it was thought that inflammation is quite linked with the type 4 secretion system. And the type 4 secretion system, as shown here, has been crystallized by others, was known to secrete the CAG-A protein. And actually a Japanese colleague called this the first bacterial oncoprotein. Now we think that this is a very important protein. We also did some studies of those to characterize and what it's doing, but it may not be really the main factor of transformation. It helps, and it changes and manipulates the host cells after infection. However, one important aspect, which I already mentioned, of this interaction with the type 4 secretion system and epithelial cells is inflammation. So we were interested in how inflammation actually is induced. And as I mentioned, there is this type 4 secretion system, and CAG-A is translocated. After translocation, it becomes phosphorylated by host kinases. And it was thought for a long time that it's this protein which is doing this. Now we studied this in much detail by doing Rnai screens, by having a readout for NF-kappa B activation here. I cannot explain this now in detail. And by doing kinase-based screens and also genome-wide screens, we identified two novel factors, namely the ALK1 and the TIFA. They were so far not implicated in NF-kappa B signaling, and ALK1, the function of this ALK1 kinase, was entirely unknown. So with a lot of work, we found out that actually ALK1 was upstream of TIFA, and after stimulation with the type 4 secretion system, and a factor which is co-secreted together with CAG-A, namely a heptose, here heptose biphosphate, and later on we identified ADP heptose, trigger the activation of ALK1 and then TIFA, forming nice complexes here in the epithelial cells, so providing a platform for further NF-kappa B activation by the canonical pathway. So then, initially, based on also other publications, it was thought that it's HPP, and then ADP heptose was found to be about 100 times more potent compared to HPV. And this is actually ADP heptose, an intermediate of LPS synthesis required for the LPS of many gram-negative bacteria. And it seems to be released via this, in case of Helicobacter pylori, via the type 4 secretion system. So what we have learned from this is that actually the principle of NF-kappa B induction and the immediate response of an infection with Helicobacter pylori, which is actually going on already by the epithelium. We don't need an adaptive immune system. It's already innate, inbuilt response, which leads to the release of defendants and other antimicrobial factors. Now the question is, how does Helicobacter pylori cope with this response? Because it was really understood that this is a particularly strong response. And actually, there are also defensins have been identified in this response, which were active against H. pylori. So how does it actually survive? And this is a lifelong way? This was the key question which we studied in the last couple of years, and this is actually based on a finding which we had some more than 10 years ago. The identification of an enzyme in Helicobacter cells, which is called the cholesterol alpha glucosyl transferase. It leads to modification of host cell cholesterol by a glucose residue. And once Helicobacter is doing this, it's also extracting this cholesterol from the host cell membrane and incorporates it into its own outer membrane surface, probably for its own benefit, to be more resistant by any kind of targets that antimicrobial factors that could kill this bacterium. So the enzyme is shown here once more. It leads to the glucosylation of cholesterol, and thereby extraction of cholesterol occurs. This was in 2006, already quite a nice finding. We knew that this reaction is somehow involved in T-cell regulation. We had very nice mouse studies on this. However, de facto, we did not know what this function is actually good for and how it influences the immune regulation. Until we made studies with the induction of interferon gamma on epithelial cells. So we used epithelial cells, treated them with interferon gamma here in the non-infected stage, and found that STAT1, in this case, is upregulated, not only upregulated, but also phosphorylated, or being activated, only in the presence of interferon gamma. Now, when these cells were infected, this did not happen at all. And we tried the common mutants, where they would abolish this effect, but they did not. Only the mutant in this cholesterol glucosyl transferase. When we introduced this mutation, the activation of STAT1 by interferon gamma still worked. So it turned out that this factor somehow blocks the activity of interferon gamma, and also later on we found IL-22. And it's doing this by extracting the cholesterol from the host cell membrane. And it turned out to be true, people suspected this already, that these receptors, the interferon gamma receptor, but also the alpha receptor, several other receptors, and TP130, IL-22 receptors, are dependent in their function on cholesterol in the host membrane. And once this is extracted, there's no signaling going on anymore via these pathways. NF-κB signaling, on the other hand, is still going on. So this leads us to a scenario where we can see that when Helicobacter pylori infects human epithelium, as shown here in vitro on this monolayer, these cells, also neighboring cells, and also the NF-κB signaling is going through these cells are negative for phosphostat 1 and phosphostat 3. And there is no response at these particular sites. While on the other areas, probably, and we can postulate this because the mucus produced here by this epithelium is highly toxic against H. pylori. But apparently here is a site where Helicobacter can survive. This can also be nicely shown from the top. So when you look at these filters with cells on it, so there are areas which are infected with H. pylori, and when you trigger this with interferon gamma, you get nice activation at the areas of STAT1 where there is no Helicobacter pylori. At the areas where you find H. pylori, the activation of STAT1 does not occur. The same is true for STAT3. No activation here at the infected sites, and at the non-infected regions, there is actually a nice activation. So to illustrate this, we postulate or have actually identified, and we can see it in our models, that there are protected niches which are colonized, which are occupied by Helicobacter pylori. And Helicobacter pylori also, it injects ADP heptos and causes recruitment of inflammatory cells. Is protected at the site because of the extraction of cholesterol, while cells infiltrating are not capable of transferring the signal and translating this into the release of defensive factors. This is different in the close vicinity of this epithelium, where there is a strong inflammatory response going on. So only at the sites, at the tiny sites of microniches where Helicobacter pylori colonizes, there is protection. Now, this is a slide summarizing the immune escape and immune suppression activities that are exerted by Helicobacter pylori. It is known that there is immune suppression. So Helicobacter has two factors, the vacuolating cytotoxin and the gamma-glutamyl transferase, which are known to block and also alter the interaction between T cells and antigen-presenting cells, actually leading much of the response here to the regulatory cell type, which we just heard about. So Helicobacter actually induces an enormous amount of T regulatory cells and also thereby leads to immune suppressive phenotypes and also probably to the decrease of asthma, for example. So it's a nice immunosuppressive agent. However, this can be nicely circumvented by vaccination. And we have shown this by our own vaccination study in mice and also in a human trial, that we are able to induce active T cells, CD4 T cells, which in mice to some extent lead to protection. They can be induced in humans very nicely, and Novartis has shown this with their vaccine effort. However, there is no protection at all, and this is most likely due to the expression of this glucosyl transferase, which blocks the defense of the epithelium due to the blockage of signaling into the epithelial signaling cascades. So also explaining the mechanism how Helicobacter actually survives in these niches lifelong. A very neat phenomenon. So with this in mind, we thought we now could really tackle the question of what is the relationship between this Helicobacter pylori infection and cancer. And we are still working on this, and this is a topic which will be done primarily in Kiel. Just to give you some background, there are infectious agents which are accepted to be involved in cancer. Of course, numerous viruses, and here the relationship is quite clear because most of these viruses drop some of their transforming DNA in the human cells and sometimes also the genome. And it can then be nicely associated with the transforming activity. This is not the case for bacteria. Here we don't have this nice linkage, at least not yet demonstrated. However, due to the clear epidemiological relationship between H. pylori and gastric cancer, it is suspected that of all the cases here, 100% of infectious agents, so Helicobacter pylori accounts for about 35%. But there might be other bacteria and it might be a strong underestimate. That is our assumption. So to bring you back once more into this situation where we have this protected niche, now I would like to introduce you into a phenomenon which comes from the clinic. So the gastroenterologists actually see this all the time. So they look into the stomachs by gastroscopy. And once a person is infected, this usually leads to some kind of inflammation because of maybe the release of ADP heptose. And this can also lead to destruction of the tissue, cause duodenal ulcer. This is now treatable with antibiotics. So you can treat bacteria with antibiotics and then you get rid of your duodenal ulcer. It can also progress to atrophic gastritis. In most of the cases, it does. And then also in many of the cases, it leads to the formation of intestinal metaplasia. So what this means is that there is intestinal pipe of epithelium in the gastric mucosa. This is unusual because you should have acid-producing cells there. So the gastric cells apparently have been replaced by intestinal epithelial cells. Now, the interesting part of this intestinal metaplasia is that here suddenly Helicobacter pylori is cleared. So how can this happen? We never get rid of H. pylori. It stays lifelong under normal conditions. Vaccines do not work. You can activate T cells, but you don't get rid of it. So the immune system is not capable of the adaptive immune system, at least, is not capable of clearing H. pylori. So during this time of colonization, besides its inflammation and perhaps also associated with this inflammation, there is also some damage going on in these epithelial cells. So there's progressive DNA damage. There are also epigenetic changes. And from there on, actually, without H. pylori, the cells may progress to cancer by itself. So without the influence of H. pylori, probably based on the mutations which are already introduced here at this stage. We have previously shown that H. pylori clearly is capable of doing DNA damage, and probably also, and this has been also demonstrated by sequencing, generating mutations in the gastric tissue. So we know that intestinal metaplasia, there are already mutations and epigenetic changes. Now let's go to this configuration. This is the starting point where we have a chronic infection of H. pylori. We have these microniches, cholesterol extraction, and Helicobacter is safe. However, it's a dangerous situation because it might lead to rupture and actually sudden death of people. Now, this can be treated with antibiotics. I mentioned this already. So it's really a serious condition, at least for some people. What happens, we know this, is that mutations are introduced. Due to the inflammation, probably other factors are contributing to this. We don't know about the details of this for sure. However, what we speculate that some of these mutations actually elicit antimicrobial activity now, not induced from the immune system, which is outside, which is recruited to the epithelial site, but intrinsically by the epithelium itself. And such mutations might be typical precancerous mutations, and there are lists here of mutations which have been found under this condition and also at later stages, particularly mutations in p53. Now we are working on this, and I will hopefully be able to present you some data in the near future on this. And there are still a lot of question marks, of course. This is still hypothetical. However, we can speculate a little bit. What we know for sure is that there are mutations in the intestinal gastric metaplasia, and somehow this leads to protection because of this metaplastic transition. We also know that this metaplastic condition can lead to cancer in a fraction of these patients, depending probably on what kind of mutations they have and how this progresses. So the question is now, is there a causality between these mutations and protections, protection first, and then between the protection and cancer? So the defense, antimicrobial defense and cancer. If this would be the case, we might have or might postulate a third type of immune defense on top of innate responses and adaptive immunity, a genetic protection mechanism. Of course, we have to ask, can we demonstrate causality between these chronic infections and cancer? Now, this brings me now to Robert Koch and his postulates. What he claimed is you need to have four reasons to associate infections with cancer. You have to have epidemiological evidence, you have to isolate the infectious agent and get it in pure culture. You should be able to cause disease, for example, in animals, and you should re-isolate it and prove the authenticity. Yeah. So this is not so simple if you want to prove a link to cancer. Yes, we have some epidemiological evidence for Helicobacter, definitely, the Helicobacter and gastric cancer, definitely. We have it also isolated, and we can also do some studies in animals. What we can do is, and this is different to the short-term relationship, so this is the long-term relationship between infection and cancer. We can look into mechanisms and see whether and how this bacterium might be able to transform. And this is what we are doing. And I think with understanding the background of and the reasons and the logic behind the extraction of this cholesterol, allowing for chronicity, gives such mechanistic insights. Also the function analysis of functions of other virulence factors, like the CAG-A protein, might help in this regard. What would definitely be a nice advance if we could identify something what researchers have achieved for viruses, that they can detect a genetic signature which is typical of a certain pathogen and find it in the cancer cells. Now, since bacteria do not release the DNA into the host cells, this is quite difficult. However, there is damage produced by bacteria, and this damage might be somehow characteristic. It might be not completely random. It might be typical of a certain pathogen, and this signature might perhaps be found in cancer cells later on. And if we would achieve this, this would probably be nice progress. And this is what we have undertaken to investigate using bacteria which produce colibactin. And these are mainly E. coli bacteria, but also some other bacteria produce this genotoxin, of which the structure has recently been partially theoretically, because it's a quite unstable compound, deduced. So this is colibactin with two aggressive warheads, where it is capable of cross-linking DNA, and the subsequent repair, during this repair process, sometimes, somehow, it comes to double-strand breaks. Now, we have now asked whether the occurrence of these double-strand breaks in the human genome is somehow specific, if there are specific sites. And for this, we have applied a particular technology for sequencing of these double-strand breaks. I'm not going to go in detail. This was in collaboration with people at the Karolinska Institute. And we have been able to identify the sites. And what we found, that there is a certain pattern around these sites that can only be found when the cells have been treated with bacteria which were positive with this polyketide, so the colibactin, compared to bacteria which did not produce colibactin. In these cases, certain regions, namely AT-rich regions, were found to be predominant, and that was not seen in control experiments. So indicating that there is a signature, and by doing further bioinformatic analysis of this BLISS experiment, we found that the target sites are related to AT-rich regions, for example, AAA, TTT. So this somehow is the motive. And then we tried to, on the basis of this, we tried to model together with people in Barcelona, the interaction of colibactin with these regions, and we found very nice fit, and came to the proposition that was already postulated by other people, that these warheads in the colibactin can target two adenines on the opposite strands of the double-stranded DNA. So producing a very specific signature in vitro. Now we looked at this motive, the DNA damage motive of colibactin in human cells, and compared this with mutations in of human colon cancer patients, so in colon cancer cells of humans, and found exactly this signature in some of the colon cancers in a significant proportion. So it's about 20 or 25% of our colon cancers exhibited this particular damaged region. We further analyzed the cleavage sites more specifically, and looked at the sites where, after correction, mutations were implemented, and found that these were at very distinct sites in the opposite strands, mutations which can then be also found in the regions of the colon cancer patients and in the colon cells, colon cancer cells, at the -2 and or position 2 and position 5 in these cells. So confirming the presence of the action of colibactin in these human colon cells, leading or being associated with colon cancer. Now this colibactin mutational signature, which we then identified, corresponded actually with a known signature which was already established, but there was no correlate on how this might possibly be induced. So it's an established signature which is actually imprinted by colibactin and associates the infection with colibactin carrying bacteria with colon cancer. And we found that the presence or the correlation with of the mutational signature and colon cancer patients was particular for those who had cancer in this region, in the sigmoid curvature of the colon, but it might also happen in other parts of the colon. And somehow these bacteria must have access or transformed differentiated cells into stem cells. So they must have access somehow to the stem cell compartment in order to implement these mutations. And thereby, this is how DNA then is cross-linked, and by resolving this cross-link afterwards, in order to allow further propagation of these cells, somehow the mutations are introduced in a very distinct pattern. Now, I think this provides already quite nice evidence for linkage between, in this case, one particular infectious agent and human cancer in the future. And I know that many other people are working on this, and there is a community now being forming. There are other bacteria which are related to human carcinogenesis. So, for example, Chlamydia trachomatis with genital tract cancers, Fusobacterium, also colon cancer, maybe together with Colibactin, they form a kind of consortium to accelerate the growth of transformed cells. Salmonella has nicely been shown also by a previous coworker of mine to be involved in gallbladder cancer, and there is indication that it is involved in other gastrointestinal cancers. Propionibacterium, a skin bacterium considered as a commensal, might be involved in prostate, and so on. So there are multiple bacteria hypothetically involved in cancer, and this perspective really pushes us to do more research in this direction in infection oncology. So at this point, I would like to thank all my previous and current co-workers, some of whom you see here at this event. I would like to thank my collaborators, my mentors, reviewers, and advisors supporting me, the hosts and funding institutions, very important, and of course, my beloved wife and all of my family who sometimes suffered from my absence and my commitment to science and absence of real presence in the family. But before ending, I would like to just mention or get back to my idols, Robert Koch and also Max Planck. Robert Koch is opened the area of research where I was spending my life, and Max Planck pointed out the way how this should be done, namely by first having insight in order to allow application. And this is what we are, of course, we are looking at application, but I think it is very important to stick first to discovery and basic science. However, I would like to bring this one step further and should say that insight should serve to improve people's health. Thank you very much.